system, right? So I will show you something about, at least about the neutral sites that that were identified in the Synechocystis. All right. So, I got actually two questions. The first one was, what are these neutral sites? Are they exons? And the second question was, how could they be even identified? It's very simple. So you do an RNA-seq experiment. You target those sections of your genome which do not have any gene transcript. Theoretically, all of them could be neutral sites. Again, theoretically, it's not always true. There are part of the genome, they have multiple other functions, not necessarily you can determine through this just mapping the central dogma of biology. But this is the go-to reference if you would like to know about identifying neutral sites in Synechocystis 6803. This came out in 2015, and they have done just simple RNA-seq based analysis, and this is the definition. Neutral sites are genomic locations where a modification causes no noticeable phenotypic change under the relevant growth condition. That is something I would like to highlight. Relevant growth condition. Let's say I change the growth condition and I do the similar analysis, it might be different. So remember that thing, relevant growth condition. This is very important. One other question, related question I got, how big is the neutral site? So this will give you some idea how big the neutral site could be. So now look at the neutral site length. There you can have some idea what would be the length there. So if you could see, I think there are about maybe 20 neutral sites they have identified. Then they not only identify neutral sites from the genome, they also identify neutral sites from the endogenous plasmids. If you remember, I told you, in addition to the genome, in addition to the chromosome of this bug, there are 5 endogenous plasmids for this organism. So they have also found one neutral site in one of the plasmids, and then I think they have just expressed some fluorescence protein. It could be, I don't remember, either yellow or green, but that's not important. But what they have figured, for 2 different tunable promoters, they tried 3 different sites, 2 neutral sites in the genome, 1 in the endogenous plasmids, and for both of them they have got different results. So if you read this paper, you cannot get to an absolute you cannot reach to a concrete conclusion. So you cannot say out of these neutral sites, always this one will work better than the other. What neutral sites does, basically this will give you some idea about which location you can target. That's it. So that's the genesis of finding neutral sites, how we can interpret the results, and how big these neutral sites are usually be. This is just an example from Synechocystis 6803. There might be other works for other organisms. At least one we have already discussed, 7942. That's another organism for which we know the neutral sites. So again, not all the cyanos can incorporate foreign genome into their own foreign gene into their own genome. But if they could, as I mentioned, then you need to, you don't need to think about the selection pressure. If you incorporate that in that genome, you are good. So most likely the gene will be retained. I think one other question that I got is how to maintain the selection pressure. Again, this depends. If there is a work for cyanos, people have tried different antibiotics, and they have a table, they have reported that for different bug, most likely this is the range you should operate on. But then again, I can tell you from my limited experience, that is not always true. You cannot, these are just ranges. So you need to, some of the times you need to try multiple different concentrations, and these are the things which usually get tricky when you have double or triple knockouts. Basically, then it's really tricky. The selection pressure, what amount. Sometimes it's purely based on your experience. I was inexperienced, so when I went and started working with Himadri, I had 0 experience working in lab. So I tried many different things, it didn't work. I talked to one of my colleagues and she said, well, you know what, this will not work. You should have talked to me before. I did exactly the same thing, it doesn't work. So these are the concentrations, usually these work. So then I tried and then it worked. In some cyanobacteria, not cyanobacteria, the photosynthetic bacteria that I'm interested in, interestingly, it has inherent resistance against most of the antibiotics that we usually use in the lab. What are the most prevalent antibiotics we use in the lab? Ampicillin. What else? Kanamycin, gentamicin, spectrinomycin, right? So 4 or 5. So interestingly, other than ampicillin, it has got resistance for everything else that we normally use. And even for ampicillin, I have to use a very high concentration. So that is kind of interesting. This is a wild type purple non-sulfur bacterium, yet it has this resistive capability against most antibiotics that we usually use. So what I'm basically trying to tell here is that these as a system, these are fascinating systems. So the more we study them, the more we know, and maybe then we can be able to manipulate them. I am still studying this bug, so I'm not there. I built a genome scale model, but I'm not still there to engineer it. So that would be something that I'm planning to do in the next few years. All right. I had few other things to say, but I'm not sure should I say it now, because few of the questions that I set for the quiz, actually it's based on what I'll say right now, because I think as a class you need to understand at least a little bit about, you understand the flux balance analysis, that's good, but how about purely from a theoretical point of view? If I tell you like these are the fluxes or these are the reactions in a model, can you tell me which flux range is feasible or not? That concept is very important. So I will probably talk about it if I can manage some time after the second lecture. So let's see how fast we can move. But before I do that, is there any question? Good. All right. So as for the we are kind of going back and forth about the model development. So you have definitely seen me talking about models from a generic context, if you remember. I started with how you can develop a model, and then I said how you can develop a model for a photosynthetic microbe. For this lecture, I will limit myself to just cyanobacteria. This will be purely based on my work on cyanobacteria, and I worked in 2 different organisms. You already know about Synechocystis 6803, and you also heard me saying Cyanothece 51142, which can fix nitrogen, if you remember. That bug could fix nitrogen, and it fixes it. It basically separates oxygen evolution and nitrogen fixation temporarily. So it fixes nitrogen in the dark phase. So I will first talk about the importance of these 2 bugs, and then I will basically tell you how existing data resources could be used to refine the model, and then given the time that I have, I will talk about some diurnal difference that I did identify and how that could be interesting. Okay. So first, model reconstruction. So even before that, why we do care about modeling this bug, right? That is the basic question that we should always ask ourselves. So if you can build a model, why build a model for this bug? So the reason why we started building a model for this bug is, from the phylogenetic point of view, these 2 are very close. Synechocystis and Cyanothece, they are very close. The difference is one can fix nitrogen, the others cannot. So we are interested about building a model, study their gene expression profile, protein abundance profile, and see whether we can develop a mechanistic understanding why that's the case. One other goal was, can we make this bug as a nitrogen fixer, right? This is a pure and simple synthetic biology. You have 2 different systems, one can fix nitrogen, the others cannot. Can we identify how this bug can do that and build a system and transfer that system here so that it becomes a nitrogen fixer? Unfortunately, due to the time constraint, I will not be able to talk about that. I myself was not part of that experimental part of the project, but I can tell you these folks have been working since 2013, and yet now when they transfer the nitrogenous cluster, which is a huge cluster, to this bug, they were able to get, I think, 5 or 10% of activity. So it's not that great. Can you take a guess why that's the case? No, let's talk about if you are hacking a molecular machinery from one organism to the other, even though they are closely related, what could go wrong? Why do you think you may not get the desired property? I heard something, regulation. Who said that? Regulatory signals. What else?
So we also have to modify the carbon fixation system because ultimately nitrogen fixation will utilize that.
True, but I think if you think about the growth rate and carbon fixation rate, they are not that different. They are not that different. Anything else? Compatibility. By that you mean whether you can really get something from Cyanothece and clone into Synechocystis. Cloning was an issue because the plasmid was really big, but that has been taken care of. Yes.
Protein interaction, like any kind of interaction in compatibility.
Right. Okay. So based on what I know, again I left that lab 2016, so I'm not quite in touch with them, but at least to the best of my knowledge, regulatory mechanism was the bottleneck for them, because even if you hack all these things like the entire cluster which has 35 genes, it's a huge cluster. Even in the cluster there are regulons, so they claim that they got everything and they have transformed into Synechocystis, but I believe the regulatory mechanism has not been fully captured, which is why this is what happened. So the take home message here is, although you might see these two systems which are very close and you are basically doing a pure and simple synthetic biology experiments, right? You got all these 30 something genes which are in a cluster, you took them, you put them in a plasmid and you transform them, and now they are part of your genome, yet you only get like 5 to 10% of the activities of all the proteins which are involved. So again, these are the problems which are fascinating. Why? Because if we could understand how a microorganism fixes nitrogen, if we could understand that, the more we understand, then it's possible to engineer a microbe so that it can fix nitrogen. At the end of the day, the long term plan of this project was, and you might laugh, that for the plant, many of the plant, we need to provide nitrogen from outside, right, through fertilizer. Can we down the line, can we make plant as nitrogen fixer? That was the long term goal, but you cannot just start from a plant, right? You start from small, you start from a unicellular microbe, you start understanding what's going on, and if you can develop an understanding, then and only then you can transfer that knowledge. So what I'm basically telling you is, like, systems biology, the possibility is huge, and the same goes for synthetic biology, but some of the times you will face difficulties, and that's when you have to understand why this is happening. But anyway, I went too far from my scope. So again, what I was doing, I was plain and simple trying to develop model for these two bugs and see how these two are different. That was my objective. So at least you have tried to build a model in K-base, right? And you have definitely you have seen some version of this slide. So model building again started from finding out the list of genes, finding out the list of reactions, and then what we did, we constructed the biomass equation based on the macromolecules. Once we do that, then we establish the GPR relationship, we develop the stoichiometric matrix, and then we fill the gaps, and then we analyzed. So I will quickly go through these steps and the reason why I am doing it, because I am sure that by now you know about these steps, right? Is there any confusion on these steps? No. Good. So, what is happening, at least for the Cyanothece model, when we were building the model, if you remember, this is a bug which can fix nitrogen, right? So, this bug's behavior in the light and the dark phase is different. So, in the light phase, as I said, obviously you could guess carbon fixation occurs, electron transport chain is active, it produces glycogen. Oh, one other thing that I forgot to mention, these two bugs have significant difference in the glycogen storage. Do you take a guess why that is the case? One stored more glycogen and in huge amount than the other. Exactly. So, that is kind of expected. So, anyway, going back to this bug. So, it produces glycogen in the light phase and then uses glycogen to power its metabolism in the dark phase, produces cyanophycin as a nitrogen sink, which it uses in the light phase. So, Himadri's lab did this experiment, RNA-seq experiment in 2008. So, they have basically developed co-regulation networks for this organism. What I basically did, I took the co-regulation network, I know which of these genes are co-regulated. They also have measured different version of biomass, and what I basically did, I used this information as regulatory information in my model. So, I have two different version of the biomass, obviously, because in the light and the dark the biomass will be different, right? And I have this information about the not the flux actually, but at least the gene expression level. So, I made a simple assumption, more gene expression, more flux. By doing this simple assumption, I basically developed two versions of my model. One can operate in the light phase, the other one can operate in the dark phase. And what I have seen, purely based on FBA based fluxes, that yes, the model could tell me that these pathways carry more flux or carry any flux in the light phase, whereas these are the pathways that carry huge amount of flux in the dark phase, which is kind of expected. So, that basically says that most likely in the major carbon metabolic pathways, that assumption was not that bad, although as I told you before, that is not the case all the time, but at least for my case that worked quite well. Now, one of the advantage of Synechocystis 6803 was it was considered, as I said, as the E. coli of cyanobacteria. So, there are many data sources available. There are many data in terms of lethal, non-lethal genes. How many of you actually understand the concept essential, non-essential genes, right? Essential, non-essential genes, genes which are essential for the fidelity of a microorganism, and genes which are not essential. But again, we need to be very careful when we say essential and non-essential. Essentiality is also dependent on the condition, your growth condition. You need to remember that, and you need to remember all the time, because these cyanobacteria, they could operate in different conditions. So, let us say you did your experiments under light or under phototrophic condition, you did your experiments under chemotrophic condition, meaning no light, no CO2, plain and simple sugar. Maybe you did your experiment under mixotrophic condition, light and a carbon source in the form of sugar. Three of you, there are high chance, and I am absolutely sure that you will get different sets of essential and non-essential genes. So, that concept needs to be very clear to you. So, are we clear on the essential and non-essential genes? What about synthetic lethal pair or synthetic lethal genes? What is synthetic lethal even mean? Can you take a guess? So, if I say these genes are synthetic lethal, what does that mean? You have definitely read about it or heard about it, right? At least does the term sound familiar? Synthetic lethal? No? Okay. Let me ask you this question. When I say that these are the genes which are not essential, right? If I take out all of these genes, do you think the organism will survive? Why?
There are some genes we don't know the function, but still
Okay.
It may not.
Right. So, you are saying we have genes which are not characterized, they have unknown or hypothetical function. True. Anything else? Again the question was, if I take out, since those genes are not essential, how about I take all of them out? How about I mutate all of them? The question is, can that organism survive? I heard no. Yes?
It can affect the regulation of that essential
They can affect the regulation of essential
gene.
genes. Yes. Anything else? Okay. If not, let me tell, yes, you are right. We might have genes with unknown hypothetical functions. We might have genes which have some regulatory roles. When we define those essential or non-essential genes, you know how that is done, right? You take out one gene, see your organism survives or not, right? That was the experiment. When you say this gene is essential or non-essential, that is purely based on that concept. Now, so the board is not here, but that's fine. So, you might know that gene B and gene B is not essential, gene C is not essential, but if you delete gene B and C together, the couple, these two double knockouts could be essential, right? Do you see the point here? Because when you take, let us say, when you take gene B, then there might be an alternate pathway. Again, I am purely thinking from a metabolic point of view. There might be an alternate pathway, but when you are taking B and C out, you are taking both the alternate pathways. So how will the organism will survive? So again, remember this concept, essentiality or non-essentiality, that is purely based on if you are taking one gene out at a time. If you are doing it in combination, double, triple, quadruple, then obviously it is a different scenario. All those combinations, they are known as synthetic lethal. Okay. Going back to my point, for Synechocystis, fortunately we have data. There was a database, Cyanobase, which provides all the list of genes which are essential. So before we built our model, there are two other models which were built. I think one is 2010, the other one is 2012. And what we basically did, we compared our results with them. So again, one thing you need to remember is we are operating in the reaction level. The information that they have is in the gene level. So whenever you are comparing this kind of mutations, you need to interpret the results. Results from the experimental studies are based on genes or reactions. Does anyone think it's reaction? Again, the question was results from the experimental analysis. Do you think those should be based on genes or reactions? Genes. And your results from the model? Reactions. So you need to make sure that you are interpreting the results. So just to give you an idea how I developed this table. So if you look at the table, on the left hand side you have in silico results. So vertically growth, no growth, horizontally in vivo results, basically the experimental results. So the way we have to interpret is, let us say we know gene G1, G2, G3 and G4. Let us say we have 4 genes. We know that 1 and 4, these two are essential, just hypothetically assume these are non-essential. So what we needed to do, by utilizing the logical relationship GPR, we need to find out the corresponding reaction. We need to make sure that there is no flux through those reactions. So if this is the case, and I am basically removing the protein step, but you understand what I mean. So let us say G1 is involved with reaction 1 and G4 is involved with reaction 4 and 5. both, it is a multifunctional protein, let us assume. So, what I need to do, once I need to deactivate the flux to this reaction and simulate the model to produce biomass. If I see the biomass level is pretty low, and again how low is low, as a user you have to define. Can anyone take a guess how can we do that? In experimental study, what did they do? They knock out that gene and possibly the bug either didn't grow or grew a little, right. Same thing here. You first find out what is the theoretical max for biomass. One value that we have discussed, 0.9, let us say. In modeling practice, it's okay if you consider 10% of the wild type biomass as your threshold. So, if you take out a reaction and your biomass value is less than 10% of your wild type value, it is fair to assume that that reaction is essential, right. Are we clear with this? So, what I am basically discussing, how to define the threshold to determine if a reaction is essential or not. In many other studies you will see 33.33%, 1/3 of the wild type biomass. In many other study it could be 1%. So, it depends on your cutoff. For this study, let us assume that it is 10%. So, what I will do, I will deactivate this reaction and see how much biomass I get. I will deactivate 4 and 5 and then I will say how much biomass I get, and then I will compare and contrast the results, and that is what these numbers are. So, if you look at the existing numbers, growth, that means out of 36 cases, 32 were good. So, there are good, the model basically predicted quite well. What about no growth? 5 out of 7, not bad, right. What about this? Model said growth, but in actual scenario it is no growth. What about the reverse scenario? Model said no growth, but in vivo results said growth. These are quite huge, and likewise if you look at the other model that came out in 2012, that is also quite high. Now, compared to those results, if you compare our model, out of 100 we predicted 94 growth, out of 19 we predicted all of them as no growth. Although there are cases where the model has some issues. So, for instance, no growth we could not predict any of them, no growth 6 out of 100. So, once you do that, basically then you can calculate specificity and sensitivity, and that is some metric people uses all the time to get how good or bad your model is. I know that some of you have asked me that question, like how do I know how good my model is? If you have this sort of data, at least that will give you some idea. So, for my model the specificity was 0.94, sensitivity was 1 compared to the models which came out before. So, definitely mine is better. But let me ask you this question now, see this number and tell me. So, if you see the model says no growth, but in vivo result says growth, what is happening? Why the model could not predict growth? Did you understand the question? Again, we are talking about single mutation, single gene mutation. Let us say you took out one gene, you got growth. If I take that reaction out, corresponding reaction, I found no growth. My question is, why do you think that could happen? What was the reason? One at a time. Go ahead. You go ahead.
Some other gene may perform that reaction.
Some other gene can perform that reaction which the model doesn't have. Same. Anyone else? Did you all understand the answer? So, the answer was maybe there are other genes can perform the same job which the model doesn't have. So, possibly the model missed out some of the alternate pathways, so to speak, or alternate reaction that the system should have, right? Exactly. So, in real life the organism has a functionality, an alternate functionality which the model does not have. So, you have to add that. What about the reverse scenario? The reverse scenario is the model says growth, in real scenario it is no growth. How about that? The model said if I take out that gene or that specific reaction, it can grow. In real life that is not the case. What is happening? Model is missing some essential, but model already predicted growth. If it misses essential, lack of, yeah, but I'm asking what kind of information is missing. I understand some information is missing. What kind of information? But the essential pathway is there because you already got growth. You got growth in the model. So, growth, not having the growth, that is not the issue. So, what I am asking, model said growth, in real life, in experiment it is no growth.
Any other gene is reactivated offsite.
So, you are saying more of a regulatory effect, is that so?
Product of the reaction must be regulating some other gene. If the reaction is not there, then the other reaction should not start it.
Okay. So, anything else?
Model over predicts.
Model over predicts, that is true. So, it could be one of the few things I will discuss. That means your model, one common theme is your model has redundancy. So, your model has alternate pathways which in real life should not be there, or they should not be active in that condition. Again, remember, we are developing a metabolic model. It will miss some regulatory information no matter what you do. So, these are the two common pieces of information that sometimes the model misses, the regulatory information, because if you take that gene out, the gene might have some regulatory effect, or the alternate pathway that you do have in the model in real life that should not be there. So, try to understand this too, because this is something we will discuss again in the next lecture. So, to quickly recap, if you have this growth phenotype, if you have this data, you could use that data to refine the model. And just to refresh your memory, we need to refine the model to make it more predictive, because at the end, if the model is not predictive, then what is the point of developing the model, right? We would like to build the model to generate good hypothesis. A bad model will not generate good hypothesis. We all agree to that, right? Any question on this? So, I will not probably talk a whole lot about how I did it, but to fix some of the things I need to make some changes, as you said. So, I can give you one example actually. So, for the sake of this discussion, I will call no growth as NG, growth as G. So, if I say NGG, I basically means that red on the left bottom corner, basically this one, NGG, okay. So, let us discuss again what was the issue. The model said no growth, in real life we have growth. We already discussed, that means the model has missed something. Do you see that it is a gap fill kind of a problem? Do you see that parallel? So, that means that we have to add some functionality in the model so that we could fix that. Exactly that is what we have done when we developed our model. The reverse scenario, G and G. G and G is the model says growth when we take out one gene, experimentally it is no growth. That means there are some missing elements, there are some redundancy in the model. some of the times you can fix that by taking or by tweaking the GPR relationship. Why? Let me tell you. So, let us say we have a reaction. We have a reaction and we know that in this reaction we have gene 1, plain and simple, right? We could not find any other alternate pathways, so we could not fix the NGG case. What we could do, we could add an unknown component here, which I do not know, but if I could get some evidence that most likely this is not a single protein, this is part of a complex. What complex and what is the other subunit, I have no idea, but I know maybe from literature, maybe from a related organism, that it forms a complex, but I do not know what the other part is. I will just say A. So, when A is not there, then this reaction should not be active. This is a very plain and simple way you can fix some things. So, what I am basically trying to say here, different kind of fixes will be required when you are fixing your model, but at the end of the day you have to refine the model to make it more predictive. Any question? You might have remembered I talked about 13 CMFA, right? And I also talked that Amit will be talking about the details how that is done. And again, for this slide I am not going to talk about the details how this is done. What I will assume that you will learn it tomorrow, but let us assume that by doing something you can measure or estimate some of the fluxes. So, what is happening here is a group has measured or estimated fluxes through the central carbon metabolism for this bug. So, once that is done, if you look at these boxes, all these boxes, these numbers are experimental measurements. These are experimentally measured fluxes, and the ranges that you are seeing here, that is the range that you will get from the flux variability analysis, as we have done in the class. We minimized, maximized a flux, and we know that these are the possibilities. A reaction can take any of these values, but we know that given the condition, given the nutrient uptake, it will not assume any other values. The values will be in between this. So, if you look at these boxes, in most of these fluxes, they lie within the range of the flux variability based values, although there are some of the times when it lies outside, right. But if you look at them, most of the times they are in the same order of magnitude. So, now we are doing model refinement, but we are using a different set of data. First we have used the mutant data, if you have it, we refine the model. Then we have used the flux measurements, and we compared in silico flux versus estimated flux, and then we can compare. Some of the times you will see there are some differences. So, one of the major difference was oxidative pentose phosphate pathway. What is the purpose of oxidative pentose phosphate pathway? What happens in oxidative pentose phosphate pathway? Can anyone answer that question? We know about glycolysis, TCA, and this pathway. These three belong to the central carbon metabolism, right? So, what is the purpose for OPPP? Is it a pathway to produce or consume energy? Do we produce energy or do you consume energy? I heard consume, I heard produce. What is it? Who said what? Is it a pathway that is usually used to, like, break down something and produce something, or the other way around? Is it a catabolic or anabolic pathway? So, if it breaks down something, it is possible that it might be producing something. It might be producing energy, right? That is what it does. But when you are dealing with a photosynthetic microbe, what will be the major source of energy? Sunlight. And you do not see why OPP should be active in a light condition, right? In the model, we did not see that. We did not see OPP to be that active. It is active, but only it is carrying some low value of flux. When they did the experiment, they found at least 1 order of magnitude higher flux. So, that is one major difference between the model predicted results versus what they could estimate. The reason why I am mentioning all this, these are the differences that you will see. Again, at the end of the day, you are dealing with a metabolic model with the assumption of some regulatory mechanism. There is absolutely no way you could capture everything. One example is this. So, there might be something going on that as a metabolic model it could not capture, but this is a commonly known phenomena. Even the fast growing strain, if you look 2973, it has higher flux through OPPP in the light condition. So, I do not know what's happening. Okay, quickly going through the differences. So, we built two models. One can fix nitrogen, Cyanothece 51142, the others cannot. For Cyanothece 51142, we did not have the mutant data, because creating a mutant in Cyanothece 51142 is a hell of a job. No one, there are few available, but there is not a list of genes which are essential or non-essential. There is no flux data available for Cyanothece 51142, which is why what we did for Synechocystis we could not do for Cyanothece. But at the end, when we compared, we saw that what we have basically expected. So, obviously one can fix nitrogen, so that pathway is there. For the others it is not there. So, one can fix nitrogen, right? For that one, urea cycle, if it's not, if it's not complete, it's okay, right? That's what we saw. So, for Cyanothece, urea cycle is not complete. For Synechocystis, it's complete because it doesn't have a nitrogen fixation mechanism, and it has few other differences. One can produce hexadecane, the other one can produce pentadecane as alkane. So, these are the differences that we have figured, and then I believe we have used the Synechocystis model to test some product. Two products that we have tried, one is isoprene, and I have given you an example of limonene, if you remember in the first lecture. It's a similar isoprenoid, same pathway, two steps are different, and we have tried for hydrogen production, and you could see if this is your experimental yield, at least they are very close. So, this is just to show how for a given product you might be very close, but then again you have to always remember that will not be the case for everything, for all the product. Fortunately for us, it was the case. Another use of this kind of a model is how can you build a synthetic pathways, because from the modeling point of view, if you do not have a reaction, you have to add that reaction. So, what we were trying to do, how we can produce fatty alcohol from fatty acid. In one we saw only one step is missing, the other we saw two steps are missing. The difference was these steps are energy intensive. So, missing one step versus two steps will definitely affect the yield, right? If one is more energy intensive than the other, then obviously when you maximize the production level, in one you will get more than the other, and that is logical. So, that kind of experiment you can definitely do very quickly in silico if you have a model. Any question? Yeah. Oh, yeah, I mean, because maximum theoretical yield is based on the model, the scope of the model, that you always have to remember. The scope of the model, if I think about the scope or the scope, so let me tell you this, the scope of the model will always be a subset most of the times of the scope of the organism, right, because we could not capture everything. Does it make sense? All right. So, now this is what I was planning to do. You all, can you access internet from your phone? Can you? Okay. So, this is what I need you to do. So, let me see if I can access internet. Okay, so now I need you to follow these steps. Where are my steps? Okay, all right, go to this website. If you go to this website, you will see two different kinds of login: teacher login, student login. Click student login, and this is the name of our class, Saha 6609. If you select that, you will see a quiz name L7Q1. So I need all who are in this part of the class. You are group 1, you are group 2, and you are group 3. Don't input your name. I don't need to know your name. I need this survey to be anonymous, and then answer the question. If anyone has a difficulty, please raise your hand, so I'll come and talk to you. Did you?
So this is enter your name. So should I just click?
Oh, if enter your name, that would be your name. Lecture 7, comma group 3, because you are group 3. Everyone in this part of the class, you are group 3. Don't input your name. Yes. Did you? It's working.
Yeah, it's working.
So you are group 2, group 1. Yeah, G2, group 2, whatever, that's fine. Did you? Can you? Yeah. So it's just one question. It's a multiple choice question, although there are actually multiple choices. So, yeah. And then login, student login. All right. So I'll give you 2 minutes, 2 minutes only to think about and answer the question. Is there anyone finding difficulty to access? Yeah. Okay. Student login, and then six 609. Oh, typed it wrong. So you will be lecture 7, comma group 1. So this is an individual answer. Don't talk to anyone, because next I will let you talk to your partner. That's the goal of this exercise. Yeah.
There was one question and then I finished it and then now it's, is it supposed
That's it. That's it. Your job is done. I can see a live like feedback here on my. One more minute. I could only see one. Can you? Okay. Okay, 30 more seconds, let's wrap it up because then I'll let you talk to your partner. Let's do it soon. Yeah?
ইন্টারনেটে চলছে না আর এটা
Oh, so if you cannot submit your answers, can you try right now again? Is there any problem in submitting the results? No? Yeah. Okay. Let's. All right. So next, I already got some of your answers at least. What I need you to do, I need you to talk to your partners right now. Now you talk to each other and see whether you can come to a consensus, whether you can come to a consensus about the answers. Go, 1 minute. Okay, 30 more seconds. What will happen? You will finish the discussion and then you go back and answer the same question again. So basically, I would like to know if you are changing your answer once you are discussing with your partner. So 30 more seconds. I should not probably show you how the answers are changing. Okay, go ahead and input your answer again. Are we all done? Still some of you are working. Can I see your hands who are working? One. Who else? Two. Okay. Let's wrap it up in next 10 seconds. It doesn't really matter what you pick. You do your best because now I will talk about them. Are we all done? Who is still working on it? All right. Okay. If any of you is working on it, I'm finishing the quiz. You will not be able to submit the quiz. Okay? All right. So what do you think the answer is? Who among you thought about a different answer but changed your answer after the discussion, and why? Yes, go ahead. What did you pick before and what did you pick after, and why? Gap filling. So can we just repeat the question for the sake of the class? What was the question? How can you refine a metabolic model? So the first option was what? Gap filling. Yeah, so that was our first answer. And then what did you switch to? Change to what? Which one? ABE. Okay. Anyone else who has a different answer before and different answer after? Yeah. Okay. Do you remember what was like ABE? One is gap fill, B is, and E? Okay. All right. All these are important, but the reason why it's H, because all the others are also important. Given the data you have, if you have those data, you can definitely use them to refine the model. Yeah. So growth phenotype is basically growth, no growth. If you do a gene knockout study, that kind of stuff. All right. Again, as I said, this is for me. I just downloaded the data. I'll see how it changed, and I don't need to know your name. As I said, that was not the point. All right. So are we good about model refinement? I think I have 2 more slides. I will get them done, and then we'll take a break. Yeah. What basis? So as I said, when someone determines if it's a growth impaired mutant or not, that is a user defined property, even for when someone measured it in an experimental setup, right? So in the wild type, you have a growth rate, right? You know how fast it should grow. As a user, how can you define that it's a growth impaired mutant or not? Do you decide like if it's like 10% of the wild type? Do you decide if it's 1/3 of the wild type, or do you decide if it's 1% of the wild type? As a user, you have the independence, but these are the few metrics people use. All right. Now, in the next few minutes, I'll be talking about how we can use the model to come up with predictions. Yeah? Yeah, because I'll post another one at the end of this. Okay. So isoprene, I will not talk a whole lot because I talked about limonene, and as I said, this is the same pathway that we are talking about. I don't know why it's not working. So this is the same pathway that we are talking about as limonene. So if you remember, I told you that if you don't want to find out the bottleneck reaction, but want to just upregulate a gene which basically catalyzes the terminal step, most of the times you will not be able to successful. You need to find out the bottlenecks, and then you have to target those, and then and only then you could be successful. So for the isoprene, if you remember, so for the limonene, if you remember, we upregulate this gene and this gene so that we can push more flux through this direction, and then we upregulate a gene which belongs to the second last step, pushing more flux, pulling more flux, but these are based on model predictions. You don't need to have an expert knowledge for that, if you remember, unlike the yeast example. And you might remember, one of the beauty of this modeling is you can come up with alternate design. So one design that we have discussed is you can push more flux, right? One other design is, now remember, this is the Calvin-Benson cycle, this whole list of reactions. So this is a branch point basically. So carbon get fixed and it goes up to the Calvin-Benson cycle, or it could go down to the TCA. TCA cycle is a mechanism to produce NADPH or NADH for some other organisms. So if you push more flux towards Calvin-Benson cycle, and lower flux through TCA, there is a chance that you can be able to push more flux to this direction. So by doing this design, we can also design some of the mutants. So the take home message here is, by using the model, by using these techniques, you can come up with multiple designs. Then as an experimental biologist, you can determine which one of the designs you would like to try first. Does it make sense? So similar concept for alkane. So alkane could be produced in Synechocystis. Now the question is, what are these steps? These steps are basically two steps from the fatty acid pathway. So as I said, what people have done, they try to upregulate two genes which are like one or two steps from the alkane pathway, and it was not successful. So what we did, we tried to push more flux through this direction, right? What we did, we upregulate some of the genes here, or we downregulate some of the genes here, because this is a competing pathway. If we downregulate the competing pathway, push more flux to where you expect, then you can reach to your desired yield. So these are one of the many different ways you can design a microbe so that you can get to your goal. But again, at the end of the day, you might come up with 10 different designs, maybe 3 of them, 4 of them will be useful. But a model or a modeling technique will help you to come up with these many different designs. Any question on the common theme? I know that I went very quickly, but what I need you to take from these 4 slides is, yes, you see the point of designing a microbe. You see why this could be useful, because from the logical point of view, it makes sense, because you are producing a product, you would like to make sure that you can debottleneck some of the steps. If you can do that, there will be more flow. There will be more carbon flow towards your product, there will be more product. Does it make sense? Is there any confusion? You can draw a parallel to all this to what I showed in the first lecture. Plain and simple, this is exactly like this. The only difference is, some of the examples that I showed in my first lecture was based on expert knowledge. It's not based on expert knowledge, it's based on your systems biology concept. It's a model driven prediction. All right. So if you remember, I mentioned about diurnal rhythm, right? I said that this is a photosynthetic organism. It shows different behavior under light and dark. So that's what we did. We did microarray experiment in 2 cycles of light dark. And let me show you why we did that. So when you go and look into literature, right, you probably find a lot of omics work, right? Either microarray, RNA-seq, some of the time proteome. Most of the times you will see that people will do correlation based study, how genes are correlated. These are good, I am not criticizing them, but at the end it does not help us to develop a mechanistic understanding. We do not know how all this is correlated to the end product. We know that how all these genes are co-regulated, right, but how that translates to more flux or less flux, so to speak. So that is why we did this study. So we measured the gene expression over light dark circle, and then we have our own model. What we were asking, can we predict how these two are related? Can we translate from genotype to phenotype? Because that is a huge issue these days. You will always hear about this, like can you make a prediction of phenotype from the genotype? So that's what we were doing for this project. So going back, what we did, we measured, based on our measurements, you could see some of the genes are being upregulated in the light phase and downregulated in the dark. Some of the genes have the opposite profiles. So obviously the genes which are involved in carbon fixation, the genes which are involved in electron transport chain, you always expect them to be upregulated in the light phase. What we found interesting was all these genes, they peak at the middle of the dark period. So if L1 is 1 hour after light, we saw that most of these genes, they peaked at L5 or L7, and then they usually go down. That was kind of interesting. What we also found interesting was the genes which upregulate in the dark, they upregulate at the end of the dark period. Again, if D1 is 1 hour after dark, we saw that most of those genes, they upregulate at D9 or D11. So that's what we found interesting as well. So just to give you an idea, so again, I will not explain the experimental procedure. Many of you are already doing it, or you can deduce that from literature. This is basically what I meant. So these are the number of genes which showed this kind of cyclic behavior, light dark, and you could see majority of these genes which showed cyclic behavior had a peak at L1 or L3. Majority of the genes which showed cyclic behavior at the dark, they have a peak at D11, and we found that this is really, really interesting. And then we basically went on and we found what are these genes, what are the pathways that they do belong. And for the light genes, you can see they are the usual suspects. They are from energy metabolism, they are from photosynthesis, they are from biosynthesis of cofactors, whereas when we look into the dark peaking genes, they are from all over the places. Some of them are regulatory genes, some of them are involved in the transport mechanism. So we also have developed this co-regulatory network, and we have found that basically they are two segregated network which basically cycles between light and dark. So you might see that they by changing the color, you could see, like, they are really two separate network of light peaking and dark peaking genes. Any question? Photosystem 1, what do you expect? The genes will be upregulated in light, and you could see they are peaking here at the beginning of the light period, right? Like photosystems 2, maybe the same. There are one or few exceptions. This is what I would like to talk a little bit, membrane transport. By membrane transport, basically we mean transport related to all possible metals and some of the inorganic compounds that it needs. What we found interesting, that manganese, magnesium, all these transition metals which are part of the photosynthetic machineries, these transporters always are upregulated at the end of the dark period. Can you take a guess why? So what I am basically asking, why these genes which are involved in the transport processes, why do they upregulate at the end of the dark period? So what does a photosynthetic microbe do? As soon as light is available, it would like to use that, right? Our working hypothesis was, maybe, just maybe, it's making sure that its machinery is ready, so that as soon as light is available, it would like to use that. So that was our working hypothesis. Does it make sense a little bit more? So we didn't stop our analysis on the gene expression level. We used, you have a question? Yeah, go ahead.
When does it know that it will be the end of dark phase?
Well, how does it know? Okay, that's a very good question. We trained it for 7 days. We trained the microbe, we put 12, 12, 12, 12, 12, 12. So then it could guess, well, once the 12 hours of dark is done, then it will be light. So it depends on the training, right? If you train your microbe like 18.6, 18.6, 18.6, then it will assume that. They are not that dumb, they are pretty smart. So, but that is a good question. As I said, we didn't stop ourselves just to analyze the gene expression. We used our model, and if you look at these circles, they basically represent the flux and the gene expression for a specific pathway, not the reaction, but the pathway. So you see transport, photosystem 1 and 2, glycogen metabolism. Most of the times, although the sizes are different, they correlate quite well. That means if there are gene expression in a pathway, there are reactions, fluxes in the pathway. So, they are most of the time they are correlated. So, that is the reason why we did this study. So, can we really translate the gene expression behavior and translate them in terms of reaction flux by using our model. So, that was the goal for this study. So, I had one other activity, but I am not sure if you would like to do it now, take a break and then continue for the second lecture. Do you have the energy to do it now, or we cannot, like, we are fine if we don't do it, but up to you. What do you want to do? You want to do it? Good. Okay. Same thing. Now it's a different quiz. L7Q2. Second question. So, but the process is the same. Okay. So, now you will be able to answer. Yeah. 6609. Sorry. Saha 6609. So, do not talk to anyone to answer the question, because that basically fails my purpose. Answer it whatever you think, and then I will let you talk to your partners. So, let us wrap it up in the next 30 seconds. Is anyone still working on it? No? I will close the quiz if you are done. Again, is there anyone who is working on it? No? Good. Now, go and talk to your partners. So, let us talk for 1 minute, because we are running out of time. 1 minute, and then we will answer the question again.
just about the software thing. It's all about all of the our knowledge that we previously have.
Right.
It's not always about the gene expression. It's also about the more knowledge apart from genes.
Yeah.
expression.
Yes. Yes.
Can
True.
I-
Yeah. Yeah. You are not talking to anyone? All right, let's input our answers again. Same process, same class, 6609.
What is the percentage of right?
I usually don't care what's the percentage. I cannot see what's the, because like when I designed the quiz, I didn't input the right answer. I didn't, because as I said, I mean, I'm interested how many of you are changing, why you are changing. If we can discuss those cases, like those of you who haven't, like who knew the answer and then you know that that's the right answer, probably will not learn anything interesting from them. But especially for the folks, it's a very common technique that I use in big class actually, because in big class, this is not a big class, but let's say a class of 100 plus students, I cannot go and interact to all of you. Even I cannot have eye contacts, right? So I have to get people's attention. This is one of the ways I can do that. All right, let's wrap it up in next 30 seconds, and then we'll discuss about the answer, what I think should be the answer, and then you can argue if you think otherwise. Is there anyone else who is still working on it?
Yes, sir.
Okay. Can we wrap it up in next 10 seconds? Yeah? Is there anyone else working on it? All right, then I'm closing the quiz. Okay, let's talk. What was the question? What was the question again?
Model-driven engineering approach always give non-intuitive.
Okay. So the question was, model driven engineering approaches will give us non-intuitive solutions and what?
Regardless of type of organisms.
Yeah. So I could see, like I didn't really see all the answers, but I could see more reds once you have the discussion. Like previously before the discussion, it was like green and red. Green means yes, true, and red means false. But after the discussion, I saw more reds. So again, I will ask for volunteer who has thought differently before and after.
Type, quality, state of knowledge base. What is that?
Okay, so knowledge base is basically very broad term, right? Knowledge base could be knowledge about mutant, knowledge base could be knowledge about fluxes and everything. If you have a good annotation, that is also part of a knowledge base.
State of the knowledge base.
State of the knowledge base is basically, for instance, let's say you are building a model on 6803. I am building a model on 2973, which is the fastest growing strain. Which knowledge base you think would be of higher quality? Yours, right? Because that's the well characterized bug. People have been working on it for decades. So your knowledge base will obviously of higher quality than mine. That's what I mean by that. Again, so what do you think, like, can I know if there is anyone who changed the answer before and after? And just share with us why you did that. There is no shame here, because you will not be judged based on that, so that we can, I mean, the whole purpose is so that we can have a discussion. Who has done that? I know some of you have done that. I know that. I don't know who, because it's an anonymous survey, but I'm asking for a volunteer. Is there anyone who did that? Okay, if you are that shy, so let's talk about it. What was the question again? Model driven approach will give us non-intuitive solution based on
And it is regardless of organisms.
regardless of the state of the knowledge base, right? And I saw most of the answers were false, and that is the right answer. It's false, because model, how good or bad it is, depends on the knowledge base. We also talked about it exhaustively. So if that's the case, then we could have built model for any organism. We don't care about the knowledge base. That's not the case. The whole point of this class, I'll come to, is to refine the model, use the knowledge base, refine it, so that the model becomes more predictive. So that is the right answer. You are smiling. Why? Okay. What was your question?
I actually took two because
Yeah.
I thought it to be like the question was building a model will give us in any way something which is non-intuitive.
Right.
So isn't it like building a model will always give us non-intuitive irrespective of the case study? Like it won't be case specific.
Okay, I understand. What you thought, and in the way you thought about it, that's right. Yes, a model will most likely give you non-intuitive solution. But when I say non-intuitive solution, probably I could have made the question a bit more clearer. I probably mean non-intuitive but good solution, not garbage solution. Non-intuitive solutions but good solution. What do I mean by that? Non-intuitive solutions which are more plausible. So if you have a good model, then and only then you will get non-intuitive solutions but plausible solution. Maybe those are possible, you can test. But if you have a model of lower quality, yes, you will get non-intuitive solutions, but most of the time those will not be plausible, right? All right, so let's take a 5 minutes break. Yeah.
Sir, if the question ended with non-intuitive solution and then it said regardless the case quality,
Yeah.
and so the question format itself says it is true.
I know that some of the things I really rushed through, but the reason why I did that, because we already have discussed about non-intuitive solutions and how can you do that. I have given you multiple different case studies. My goal is if you could understand one of them and the basic principle, you are good. You don't need to understand all of them in the same time, but as long as you understand the basic philosophy, you will be fine. Any question on the last class? Right. So this lecture will be kind of an extension of some of the things that you already have seen in the last class. So we all understood that we need to make metabolic models more accurate, right? Accuracy is correlated to the predictability. More accurate model will most likely be more predictive, right? We don't have any confusion on that, right? So what we will discuss in this class is how we can use the growth, no growth data. I told you how I did use that, but I didn't tell you in details how that could be done. And then we'll talk about how you can get rid of the cycles, and finally I will give you a very quick overview of some other modeling techniques that are out there, which are not flux balance analysis based, which is different. So we will talk about them. All right. So you might remember I mentioned about group contribution method. Group contribution method is a method to calculate the delta G value, difference of Gibbs free energy value for a given compound. If you know the Gibbs free energy value for a given compound, then you can determine the Gibbs free energy of the given reaction. So the way it works is it is based on a macromolecular substructure. So if you are dealing with this specific compound, look at the boundary. The boundary on the left, that is the wrong one, the one on the right, that is the right one. Why? For that you need to understand few different concepts which probably I will not be able to capture. But what I would like to recommend is go ahead and read that paper, and you already have the slides, you do not have to write anything down, you have the slides, you should have the. So, basically the way it will work is, if you look at this equation, if I know, so let us do this exercise. If I can break my compound down into multiple different fractions, and if I can estimate the delta G of those fractions by using this simple summation rule, I can find out the delta G value of formation of my compound. Now, when I have the delta G formation of all the compounds involved in my reaction, I will consider what is in the product minus what is in the reactant, and then I will see what is my delta G look like, right. It will be either positive, it could be negative, or it could be a range. If it is positive, we know what is the deal, right. The reaction flow would be in the reverse direction. If it is negative, it is a spontaneous reaction, reactant to product. If it has both positive negative, it could be a reversible reaction, plain and simple. Many times this will not work. Why? Because at the end this is still an estimation. So we are estimating the delta G formation for a compound, and based on that we are estimating the delta G for the reaction, and we also need to have the concentration of the corresponding fraction or the corresponding compound. And many times in the intracellular environment we cannot measure the concentration, right. Like if I give you a Synechocystis 6803 cell, if I ask you to grow and culture and like subculture for few days, and then can you come back and tell me, well, I know for sure metabolite A has a concentration like this, probably you cannot, but we will have some idea about the concentration range. Many times if you follow this kind of an approach, and if you follow a general rule about the concentration in the millimole or micromolar level, you will get majority of your reactions as reversible reaction. So if I am a model builder, I am going through this pain, and after like 7-10 days I have a list of reactions, let us say 1000, and all of them are reversible. What is the point? There is no point actually. So I would still recommend to look into literature to find out this delta G information rather than using this group contribution. But this is something I would like you to know, that this kind of mechanism is there. As long as you have some information about the concentration of the metabolites, this will be better. I would not say this will be fine, but this will be better. But if you do not have that information, if you are assuming the concentration range, then probably you will not get anything good out of it. I know it does not make that much of sense, but it is what it is. So there is a tool which can do all these things for you automatically, and it's called equilibrator. So if you look for equilibrator, you can find the database. There you can just plug in the reaction of your interest, and it can predict these values for you. But again, you need to understand it will be based on the group contribution method. Okay. Next, what we have just discussed few minutes back, right? You saw that in my model I had some issues. I had some issues here, NGG, and I have some issues GNG, right? The question that we have discussed, the issue that we have discussed is, if it is NGG, that means the model is missing some functionality, right? If it is GNG, the model has redundant functionalities. So we know these are the issues. The question is, how can we get rid of them? We have discussed, if the model has NGG, you will add functionalities. When you do that, you are moving some of these things up, right? Are we all on the same page? What do I mean by this arrow? Let us say you have a mutant which can grow, and you are fixing it in your model. As soon as you fix, from this cell it will move there, right? If you can fix it, it will be GG, right? What about this? When you have issues like this, the model says growth, experiment says otherwise. So, basically you want to push it here by doing some sort of suppression. It could be a regulation saying that in that given condition the reaction does not carry flux, or it could be taking that reaction completely out, one of these two. But at the end you are moving some of the cases from this cell to this cell. So, at the end we would like this to make as NG. Are we clear on that? What is the confusion? Are you clear about this or none of them? So, what I am basically saying, let us say for a specific gene, right, you know that gene, if you take that gene out, experimentally your organism can grow, but your model shows no growth. So, that means your model is missing some functionalities. Your model might miss a reaction. You can add the reaction. As soon as you add the reaction, then the model will grow. So, that specific case which was inside this cell, it will move up, right, because then it becomes a growth condition. And similarly for this, so you have an NGG condition, you will suppress some reaction and then it will become NG. Are we good now? Is there any other confusion? So, one thing, this is the take home message here. While you are doing all this, what is the base minimum you have to make sure? Yes, you are moving things up, you are moving things down, these are in the right direction, right. But let us say when you are doing this, you are moving things in the wrong cell, then that is not helping you, right. What can happen when, so let me ask this question, okay. When you are fixing NGG, you are adding reaction. You might be able to fix that case, but what could go wrong? The question is, for a single gene, experiment shows growth, model shows no growth, which you would like to fix by adding another gene. You fix that condition. What could go wrong? Can anything go wrong?
The results are not reproducible.
Sorry?
The results are not consistent or reproducible.
No, the results are not reproducible or consistent, we know that, which is why we are fixing it.
The other cells can be affected because of that.
And I saw another hand. Did you, what is your idea?
It may go to the other wrong cell, growth, non-growth.
Exactly. So, for the rest of the class, if you add a gene, that means you are fixing something, that is what you are thinking, right. But by doing that, you might introduce an alternate pathway, an alternate reaction which should not be there to begin with. So, you can increase a number here, right. Likewise, can you think about any issue when you are suppressing a gene or a reaction so that you can move it here, what could go wrong?
The other reaction
Some
can
other
also be,
gene may be not like
That
that
gene can be involved in other reactions also.
And some other cases might become no growth, which should not be no growth, right. So, whenever the bottom line here is, whenever you are refining the model, you cannot just think about that gene and that gene only, or in other words, not that reaction or that reaction only. You need to understand what will be the overall impact on the model. By refine, okay, if we can refine without affecting other cases, we are good. But when you are refining, if you are affecting the good cases, then it may not be good. Let us say to fix one such case, you are screwing 10 other cases, good cases, then should you or should you not do it? The answer is no, you probably should not do it. So, What I am trying to say here, there is a way of doing it automatically. The name of the tool is called Grow Match. I am not sure if it is in there in the K base, but whoever has interest, it is there. That is good. So, if it is there, then you can make use of it. If it is not there, or if you are interested to use Cobra Pi, it is there. So, again the name of the tool is Grow Match. Grow Match basically does this, because we do not need to, at the beginning we do not need to get worried about GG and GNGs. It will fix this condition, then it will fix this condition, but each of the time whenever it fixes, it checks if it is like affecting any of the good ones, and that is how it does that. Any question? People at the back, are you fine? Any question? Are you fine?
Sir, how can it check the in vivo?
No, in vivo is defined. In vivo is defined. What I am saying, because in vivo results you have done, or someone else has done the experiment, you have the results. It is just based on the in vivo, how can you refine? So, how can you have the best refined model? No model is perfect, right? So, what I am saying, based on the in vivo results, what is the refinement that you could make given the data set that you have? Okay. We have talked about gap filling, right? But I have not really told you how gap filling works, right? I have not talked about, I told you that a metabolic network model have gaps, and we want to fill the gaps, but I really didn't explain how gap filling works. Gap filling works based on a metabolite. Gap filling does not work based on a reaction. Can anyone take a guess why? Again, to recap, gap filling works based on fixing a metabolite, not on fixing a reaction. My question is why?
It is all connected.
All connected. You are saying something?
Reactions of the pipes and
Reactions of pipes, they are the joints, yes. Anything else?
Nodes are the metabolites.
Nodes are the metabolites, yes.
Define the nodes to connect the.
You have to define the nodes. Anything else?
One node can be related to many reactions, like many.
Exactly. Exactly. So, what we are discussing, how gap filling works. We know that gaps are basically due to uncharacterized proteins, hypothetical proteins, some unknown information. But if we take a metabolic centric approach, by fixing the metabolite, you will be able to fix maybe more than one reaction, because at least a metabolite participates in 2 reactions, and most of the times they participate in more than 2. So, fixing one metabolite will definitely let you fixing more reactions. Think from a simple network based approach. So, that is why gap filling works on a per metabolite basis. So, we would like to fix a metabolite so that it could be either produced or consumed, because we know in this metabolic network a metabolite has to have at least 2 reactions, one that can produce, one that can consume. Does it make sense? If it is an end metabolite, then you have to define a sink reaction. So, gap fill will suggest that define a sink reaction, that it will suggest. So, again that is also based on some bi-level formulation, as we have discussed in the OPNOC. So, basically I am not showing you the formulation, but the way it works, it works based on a parsimony principle, meaning that we have a metabolic model and we would like to use minimal addition to fix it. So, let us say you are fixing a metabolite, you came up with a solution, like you can tell me like I can add 2 reactions and then I can fix the metabolite. He came up with a solution which require 5 reactions. So, which one we can pick? 2. So, usually we go for a minimalistic approach. What gap fill does, it gives you a list of solution, it gives you a list of many different possibilities. 2 versus 5, usually we pick 2, but can anyone say, or can anyone think about a case when we will pick 5 but not 2? So, what I am basically asking, you are trying to fix one metabolite. Gap fill has suggested 2 solutions. One would require 2 reactions, the other would require 5 reactions. In many cases, the majority of the cases, we will pick the one with 2 reactions. However, there are cases when we have to pick the 5 reaction one. My question is when and why? Doesn't have the resources, but when gap fill has suggested something, gap fill has checked.
Ultimately, we want to maximize.
Sorry, go ahead. One, you are saying one can produce a cycle, then it would be a problem. Go ahead.
Ultimately, we want to maximize V product or V biomass. So we have to check which of that, and one can be energy exhausted, more energy exhausted.
Yes. So you both are, or all 3 of you are talking about different aspects. But is the class good on the question, like what I am asking? Is there any confusion on that? We will discuss about the solution, but the question, is it clear? So all of you are kind of right, definitely, because 2 reaction, 5 reaction. Let us say my 2 reaction, if I add those 2 reactions, it will create infeasible cycles, then I will not add them. But the current formulation of gap fill will not check it for you, unfortunately. That is one thing. And you are talking about what is my objective, right? So what she is saying, let us say by adding 2 reactions, I will not be able to get the production level that high, which I can do for 5. Then again, you have to pick your production level as your objective. What is the flux you would like to maximize by fixing this gap? If you can define it like this, then that is out of the question. So many times that is the issue that you will face. Your main model does not have any infeasible cycle, right? It does not, because it has some gaps, it has some other issues. You found out two reaction which can fix it, but it will create a cycle, then you better not include it. If you include it, your predictions will not be good. So you better then go for that 5 reaction solution. So again, what I am basically telling you, there are automated way, but it will always require a manual oversight. There is no tool pipeline as such which will get you from start to finish a very refined model, unfortunately. So I kind of briefly talked about it. So like gap find, gap fill, there is also a tool called loopless FBA. Again, the principle is the same as we have discussed. Kirchhoff's second law in the circuit. We know that at a steady state, through a cycle, there will not be any current flow. Same thing here. So we need to make sure that is the case. So whoever is interested, I deliberately am not showing any mathematical formulation here, but I know that some of you are interested because some of you have, like, I don't know, maybe you are interested about learning optimization. So I would definitely recommend you to go and check that reference. But what it basically does, this is not a model refinement. Let me make it very clear. This is not a model refinement. This is like you build a model, you suspect that there might be loops. If you suspect that, as a checking measure, I would like to make sure, I would like to make some predictions, but I would like to make some predictions by making sure that there is no flux through these loops. Then and only then this could be useful. Again, this will not refine the model for you, this will just make sure that there is no flux through those cycles. Does it make sense? Previous steps that we have discussed before, the gap find, gap fill, grow match, all these are model refinement. You do all these things, you develop a predictive model, but you might still anticipate some of the reactions that can participate in a cycle. If that is the case, then you have to run loopless FBA, not FBA. Not sure if K-base has it, but Cobra Pi, that Python based Cobra tool, it has loopless FBA. You can do that in there. So, there are many other tools in predicting flux when you are interested about a specific product, and I will tell you I think 3 or 4 different examples. So, okay. So far what we have seen for flux balance analysis, we pick a reaction flux, we maximize, right. That is what you did, that is what you saw me doing, and that is it, right. There are many other ways you can do this kind of maximization. So, let us look at these green lines in the spider plots. Okay. So, this is something that you will get if you do MoMA, minimization of metabolic adjustment. What does that mean? So, the assumption was, let us say you have a wild type strain and you are designing a mutant. The assumption was maybe the mutant would like to operate very close to the wild type state. So, the difference of fluxes of all the reactions, if you take the reaction flux of individual reactions, it would like to minimize the difference. We all know the least square method, right? We know that, right? So, this is kind of that concept. So, we have that wild type flux, you have your mutant flux. I would like to use that least square concept here. That is plain and simple, MoMA. So, minimization of metabolic adjustment. I would like to minimize how much adjustment I need to make. If you do that, then probably you will get this green lines basically which shows how much of lactic acid, acetate, succinic acid, threonine or naringenin I can. So, these are the targeted products for this example, how much I could get from a specific mutant. Well, the color scheme is horrible on the screen, but maybe you can see the TV screen there. So, there is a brown line there that basically shows what is the maximum yield. Let me. So, the brown line here basically, that is the maximum yield, if you remember theoretical maximum, we all understand that, right. So, let us say you are maximizing the production of format, this is the point you will get ethanol, you will get this lactic acid, succinic acid, malate. Plain and simple, flux balance analysis, we are finding out what is the maximum theoretical yield. So, we have discussed MoMA, we have discussed plain and simple FBA. Any question? And then, sorry, my bad, this is plain and simple FBA. The brown line is maximum yield. I was not thinking. Yield is obviously based on how much nutrient you are uptaken. So, maximum FBA would not consider the yield, it will only consider what is the maximum flux. So, do you see how these two are different, yield versus flux? How these two are different? How do you define yield? Yield is based on what? Biomass production, how much nutrient you are taking, maybe based on the limiting nutrient, right. So, by yield what I mean, let us take an example. How much succinic acid per glucose, that is yield. If I do simple FBA, I am interested about how much succinic acid, that is it, that is the difference. But depending on what is your objective, you can map different space. So, we talked about MoMA, yield and maximum flux. You can adopt many different objective functions as you wish. In many different applications, one is better than the other. At the moment, with not knowing the system, not knowing your product, it is very hard to say which one is better than the other. Any question? All right. So, this is some suggestion, like for a photosynthetic bacteria, what are the different objective functions we can test. It could be like maximize photosynthetic efficiency. That is most of the times under the light phase, at least at the beginning of the light phase, that is true. It could be obviously the maximization of biomass, minimization of the sum of the fluxes, and performing MoMA for a mutant, and obviously the last one is pretty obvious. We would like to maximize the production level of a desired product. So, it could be one of the many things. Some of the times you want to do multi-objective optimization. It could be more than one for a given condition. So, whenever you are using the model to analyze something, that's the question that you should be asking yourself. What could be the objective function? If it's not in the exponential growth phase, then that will require some sort of reasoning, like what do you think, and what you think at the beginning may not be what you will think at the end, because maybe you will do something, you will create some mutants, you will test, it does not work, then you have to come back and refine. When I say refine, it might be a refinement of your objective function as well. So, you better know what are the possibilities, what are the different possible objective functions you can try. Does it make sense? Okay. The next two slides, let me tell you this, these are busy slides. So, I will try my best to explain it to you. So far the model that we have discussed, I told you multiple times, these are metabolic models, right. Now, this is a concept that Bernard Paulson at University of California, San Diego, he is known of, by the way, he is known to be the father of systems biology. So, Greg Stephanopoulos is the father of metabolic engineering, and he is the father of systems biology. But anyway, so they are trying to coin a term called ME model, M-E. M is metabolism, E is expression. So, they are trying to capture the synthesis of RNA and the synthesis of protein in this kind of modeling framework. So, hypothetically, if you can write a chemical reaction to show how RNA could be synthesized, how a protein could be synthesized or degraded, then you are basically you are still dealing with a list of reactions. What will happen for this case, those list of reactions are not metabolic reactions, right. So, that is the difference. You are dealing with now metabolism, translation, DNA charging, and transcription. So, that is why they call it as a more comprehensive model. The downside is you are dealing with more parameters, there are more unknowns, and unfortunately this kind of modeling is only possible for E. coli, for which we know a lot about the parameters that we need. For the rest of the microorganisms, if we have to build this kind of a model, the major limiting factor is the knowledge base. So, you can develop a model, you might have to borrow some of the parameter values from E. coli. Now, it is your decision as a user whether you want to do it. If you are dealing with a photosynthetic microbe, it probably does not make sense to borrow so many things from E. coli. So, again I would say this is still in a very nascent state, but down the line when people will do more experiments, they will measure the Km, Kcats of all these enzymes, this will be a very good modeling technique that we can use. So, that is ME model. Again, before this slide we have only talked about metabolic model. In this slide and the next one I am trying my best to give you a very quick overview of what is still out there. I am not giving you too much information because I do not think that is needed at the moment. I just need you to know the term so that whoever is interested can go and check the citation for that. So, ME model is here. Next is, so this paper came out last year. This is basically a metabolic model. What they have done, they have used some protein information. In the morning in the first class, if you remember, I told you that if you have a flux and if you know the delta G value, or if you do not, but you can get the information from literature, and then if you know if it is a reversible or irreversible reaction, you can definitely constrain the flux range, right? 0, 1000, negative 1000, 1000, like that. So, what they are doing basically, they said that if I know the concentration of the protein and the kcat value, and if I multiply that, that is the maximum flux a reaction could carry, given that is the single protein that is out there. So, they have constrained the flux based on protein information, the translation of the protein, and that is the information that they have incorporated in the metabolic model. This is still a metabolic model, but what they have shown, by using this protein resource allocation, they can simulate the light dark condition, and most of the times they have shown that it's more refined, it's an improved model than just a plain and simple metabolic model. So, just to quickly recap, in the first slide I told you about me model, which is built as a metabolic and expression model, whereas this one, it's still a metabolic model, but it incorporated the protein information as a constraint on the flux. Are we clear? Is there any confusion? All right. Now, how many of you heard about kinetic model? We started the discussion of this class by asking you to write a kinetic rate equation, if you remember, A to B, and you wrote like R1 equal to minus K1 Ca, right? This is what it is. The reason why we cannot do kinetic modeling for so many different organisms, because we do not know these K values. You can know K values for some of the reactions, but if you are dealing with 1000 reactions and so many of them are intracellular reactions, you do not know those parameters. So, what this kind of modeling does, this is called ensemble modeling, EM modeling. What it basically does, how many of you at least know the term elementary reaction, elementary step? Okay, can you explain what is the elementary step?
In a simple reaction,
Yeah.
the step which determines the rate of, like, rate determining step is the elementary reaction step.
Not really. So, let us say you have two substrates A and B producing C and D. The steps that will involve here, I am not sure at the back you could see. So, each step will happen once. A will get to the catalyst and then B will get to the catalyst. A and B will make C and D, and C and D will come out of the catalyst. Breakdown. So, A plus B giving C plus D, that is the overall reaction, and what I have just discussed, these are the elementary steps. All these elementary steps, they assume that they follow Michaelis-Menten kind of kinetics. We heard about Michaelis-Menten, right? At least you heard about Michaelis-Menten. So, in the Michaelis-Menten equation, how many parameters are there? Vmax and Km. For each of the elementary steps, let us say you have 4, you will have individual Vmax, you have individual Km. Now, you might come up with multiple different combination for all these elementary steps. Now, if you measure the concentration, as will be discussed tomorrow, it is basically a fitting problem. How can you fit your model to what K values or Vmax value, so that you can explain a behavior? That behavior could be a concentration profile, that behavior could be a concentration profile of a mutant. Again, this is kind of starting with a huge amount of different alternate models and fitting them to one set of model which can explain the behavior, which is why it is known as ensemble modeling, because you are starting with an ensemble of model and then you are restricting yourself. So, this is one of the ways you can get rid of that measuring those K's. You can still estimate based on the dynamic profile of the concentration. Does it make sense? Again, I really do not expect you to understand every single detail, but if I explain the steps, the core basic steps, do you think it does make sense? because I know that at least you heard about Michaelis-Menten, at least you have seen Michaelis-Menten. You are interested about biology, I am sure you have you have done that. So, these are the steps which are involved. The downside is it is very computationally intensive. So, if you do not have that kind of resource, this is not possible. And another information is the largest model that could be or that has been built so far is the model of an E. coli which has only 500 reactions, just to give you an idea. Now, if you think about building a model of 2000 reactions by using this method, might be difficult. So, all right. So, this is the last slide before we adjourn. What we have discussed so far? We have discussed the group contribution method that could be used to calculate delta G. You can use grow match to match the growth profile, in silico growth profile with the in vivo growth profile, right. You can use find gap fill to fix holes or fix gaps in your metabolic network, and we have discussed that we are taking a metabolite centric approach, not a reaction centric approach. This is very important. And then we have talked about loopless FBA and how can we use different objective functions for our specific problem. And finally, in the past 5 or 10 minutes, we have discussed about multiple other modeling frameworks. We have discussed about me model, kinetic model, and how you can use protein abundance information to limit the flux through your reaction. Is that all? Yes, that is it. Thank you. This is my son. Any question?